Hi everyone, in our previous video we looked into type 1 regulatory T-cell mediated immune regulation in malaria, and I think we got up to the point where we looked at 4 different mechanisms by which Tr1 cells can control immune response with, and they were illustrated in panels A to D in this diagram. I think we looked at the mechanism illustrated in panel A, which was cytokine mediated mechanisms. In this video we're going to look specifically into mechanisms in panel B and C, which are cell contact dependent mechanisms and metabolic disruptions. Firstly, in cell contact mechanisms, these type 1 regulatory T cells can also regulate immunity through direct cell-to-cell interactions. And how they do this is they essentially use surface receptors like PD1 or CTLA4 in order to bind to their corresponding molecules, which are PD-L1 and CD80 or CD86. And these receptors or these surface molecules are essentially on the APCs, which are antigen-presenting cells. And when this sort of binding occurs, it really sends an inhibitory or stop signal to the APCs, and it prevents it from essentially just overstimulating the nearby T cells. Now, so basically this physical sort of contact-based regulation limits excessive T-cell activation, and at the end of the day, it helps prevent uncontrolled inflammation. Now, in the context of malaria, this mechanism is critical for avoiding severe inflammatory reactions that could potentially damage blood vessels or other vital organs. Now, type 1 regulatory T cells can also influence the surrounding metabolic environment by expressing 2 key enzymes, and these key enzymes are CD73 and CD39. And these enzymes essentially just break down ATP, adenosine triphosphate, and it's a molecule that's usually used for cellular energy, into adenosine, which has anti-inflammatory effects. So adenosine basically acts as a suppressive signal that essentially slows down immune cell activity and prevents overproduction of inflammatory cytokines. High adenosine levels will therefore protect the tissues from collateral damage during infection, and in malaria, this pathway essentially helps reduce excessive fever or systemic inflammation, often triggered by immune responses to Plasmodium falciparum. Alright, so that's pretty much it for today. In our next video, we'll be looking into the last mechanism, which is cytolytic activity.